Super. Moving on then with the next award. Well, I know that the next award is the Young Investigator Award, which is going to Keith Gagnon this year. Not sure why the slide is not advancing. If I could have the next slide, please. Okay. Yes, thank you. The Young Investigator Award is sponsored by Mary Ann Liebert, the publisher of our journal for the Society, and we very much appreciate their support of this. This year's award goes to Keith Gagnon, and I don't want to take up any of his time from presenting us with his very exciting work, but I do just want to acknowledge the very generous support of Mary Ann Liebert and Nucleic Acid Therapeutics, and I want to encourage everybody to submit your manuscripts to the journal, which is our official journal of the OTS. So with that, Keith.
Okay, so I think this is on. I tend to talk loud, so hopefully he can normalize my voice.
It's okay.
Thanks, Art. So yeah, thank you very much to the selection committee, the whole OTS community. We have great grad students, great postdocs, and so it's really an honor to be selected for this. Okay. Let me go back. Oh, that's just my title. What I want to tell you about is RNA-guided enzymes today, and a little bit about how they fit into my career, how I feel like from my perspective they fit into nucleic acid therapeutics, and a little bit about where I think what the future might hold for RNA-guided enzymes for nucleic acid therapeutics. I just need a chalkboard, that's all I need. Right? So when I say RNA-guided enzymes, you probably think RNAi or maybe CRISPR, but I bet many of you don't think about snoRNAs unless you come from that world or you've been around for a while. BoxCD and BoxHACA snoRNPs are really the first examples we ever had of RNA-guided enzymes, and we learned a lot from them. So I'm going to talk about those first, but just some sort of thinking points as I go through this talk. I just want you to think about what have we learned from RNA-guided enzymes, how have they played a role in nucleic acid therapeutics, and what might be the future, where might they be able to contribute in the future. So if you want to think about RNA-guided enzymes, you have to go way back before RNAi to reach all the way back to the 60s, where, they were really just trying to figure out what a lot of RNA did. And there was a lot of very abundant, U-rich, small RNAs in the nucleus. And in particular, this guy Harris Bush was extracting them, sequencing them, but it still wasn't clear what they were doing. In mid to late 80s, Stu Maxwell was primarily the driver for discovering that they hybridized to ribosomal RNA. And this led to the conclusion, or solidified the conclusion, that they're probably involved in ribosome biogenesis. So from that point on, things sort of heated up, and they began to really figure out a lot of really important things about snoRNAs. There's a lot of different species. I'm going to stand down here so I can see better. What they were actually doing was they were chemically modifying nucleotides, 2' methylating or pseudouridylating them site-specifically. Stu Maxwell discovered they were processed from introns, especially in eukaryotes. And importantly, a lot of these were found in archaea. These are highly conserved RNAs found in archaea bacteria. So I joined Stu's lab. I was an undergrad. I started working in his lab in 2002, and he was a great advisor. And I figured it was an easy thing, so I stayed on as a PhD student. So I started my PhD with him, and what we really did in the lab, our approach was to reconstitute these RNPs from purified components. So these were fun times, old school biochemistry, the stuff most students try and avoid nowadays. And I these are actually my favorite gels now. How many gels do you have where you can put 50 samples and you get nucleotide resolution? Okay, this was high throughput sequencing for RNA mapping, and did a bunch of other stuff. But I remember as a young grad student, we had this really exciting piece of data, and I turned to my advisor. I say, Stu, this is going to put you on the map. Boy, I'm already on the map. And so I realized, hey, I should not only read the literature, I better read my advisor's old papers. So that was pretty funny. He was a great advisor, though. In the years I worked with him, we studied the assembly of these complexes, their structure-function relationship, and their evolution. And what we did was really just try to map the RNA-protein interactions through mutagenesis. What were the core proteins that were required for activity? I developed an affinity purification approach to give homogeneous RNP particles that we used to do some cryo-EM mapping with Susan Basurga. And then we looked really carefully at the evolution of these at the structural level. This is comparing an archaeal protein, core protein, with the human core protein. You see how closely conserved this interaction is. Okay. The crystal structure was solved after I moved on to do my postdoc, but we learned a lot of really important principles from snoRNAs. For example, the idea of a seed sequence was already around at that point. We didn't call it that, but we knew you needed about 9 to 10 nucleotide hybridization to get site-specific editing. It was always the 5th nucleotide. We knew which end the hybridization started from. We knew that that the RNA had 2 sort of domains or modules. You had an RNA, a variable guide sequence, and a very conserved protein binding domain. That these enzymes and RNAs had co-evolved. It was really difficult to mess with the interaction. And an important point was that there was these sort of emergent properties from these enzymes. The enzyme can't catalyze the reaction without the guide RNA to direct it. And the guide RNA actually can't really stay stably bound to its target long enough to, without the protein bound. And so we see this actually come about in RNAi and other systems. But in particular, this really because you only need about 9 to 10 nucleotides at base pairing. Okay. So we really haven't heard a lot about these in therapeutics. There's a gentleman, Yitao Yu, you may know. He's at Rochester. He's actively working to try and pseudouridylate stop codons, mutant stop codons, so that you get read-through and block NMD. But basically, anywhere you can put a pseudo-U or 2' methyl into an RNA and then affect its metabolism or its structure function. So this might be a useful therapeutic in the future. And I think people will start working towards this. So let me tell you about RNAi, because I was a young grad student and finishing up the snoRNA work, and I really wanted to keep working on these types of enzymes. And RNAi was just becoming big at the time. And I don't need to tell you about the discovery. Craig Mello gave a great talk at the beginning of the meeting. But it didn't take long to start unraveling the mechanism, characterizing microRNAs, argonauts, and the therapeutic potential sort of became obvious pretty quickly when people, David Corey, and Rana, some early examples where they were chemically modifying the RNA and showing that it still worked. But there was many, many people who contributed. The point was, though, that the early work from snoRNAs, the tools, and our understanding helped accelerate this sort of research. Okay, so I'm interested in RNAi. I'm snoRNAs right there in the nucleus. David Corey, I found out at the time, I bumped into one of his students at a meeting. He's working on RNAi in the nucleus. So I didn't know much about RNAi, so this might be a good fit. So I joined his lab in 2008. And one of my early projects, and I'm just going to talk about this because this was my introduction to nucleic acid chemistry, was targeting ASOs to Huntington repeat expansions. And we found that we could selectively target the mutant repeat expansion. And importantly, I learned a lot about nucleic acid chemistry. I got to play with a lot of different modifications, and that was extremely valuable for me at that time. I also had a project that many people don't know about. It was a sort of a super secret editing project. I was editing the web page, so I had to upload pictures and update publications. But these sort of pictures kept cropping up, and David pulled me off that project. There was too much off-target editing, so that was never published. So again, let me get back to the RNAi. This is what I was really interested in doing, and at the time, David was showing you could put siRNAs inside of cells. They can change transcription, they can change splicing, but no real sort of mechanism for Argonaut or biological mechanism had emerged. And so we set about really simply just to demonstrate that the machinery was there and that it could function, to sort of try and lay that to rest. And so basically, we found you can put the siRNA in the cell, gets loaded in the cytoplasm. It will stay in the cytoplasm, but we can also go to the nucleus and target things there. We now know that it does work in the nucleus, not quite as efficiently or maybe not as predictably as ASOs. And one of the cool things that we discovered was there was really no risk-loading factors in the nucleus. So that really made sense of this model. And the bottom line is that you can use siRNAs to sort of commandeer RNAi and get it to go where you want it to go, whether or not it can actually naturally goes there. And the Core Lab is still following up on some of this stuff. Okay, so Argonaut taught us a little bit about RNA-guided enzymes as well. So the cool thing about Argonaut, it was really just like the minimal RNA-guided enzyme you needed. There was no special structure other than A form. There's a little bit of a complementarity, shaped complementarity, but it's largely unstructured. Interacts through the phosphate backbone, and you could actually really heavily chemically modify this because of this fact. And so a lot of the ASO chemistries we were able to build on that and very quickly do modifications. The seed sequence is something that became recognized and became very important, and so that's where it got its name. Okay, so let me switch now to CRISPR. See how we're doing. CRISPR is this bacterial adaptive immune system, and I'm sure most of you guys know about this. You're probably aware that it was discovered and it was first published in '95. Actually, its first description goes back even further, but they didn't really know what it was. And then it took about 10 years to come up with the idea that, hey, maybe this is an adaptive immune system for bacteria. And then in 2008, Sontheimer published a paper showing that you could sort of program this system. But the rules weren't really clear. It really wasn't clear how well this would work. It didn't get extended. But then in 2012, a couple groups, including Doudna and Charpentier, probably the most famous for this, really definitively showed that you could program these enzymes. And then it was only a year later that it was demonstrated that you could knock out and edit genes inside human cells. So what's surprising really is that was only 4 years ago. So this has moved really, really quickly. And I'd like to think that it's built on the RNAi that we learned and even the early snoRNA research. Okay, CRISPR-Cas has taught us some more things, and we're still learning. There's a lot of CRISPR systems out there that we're learning about. For example, you can have 2 RNA guides, maybe more. And target binding doesn't have to be only decided by the hybridization of the guide region or the guide sequence. You can have additional sequence specificity by the protein. So I think for a while, initially, maybe people thought, and I thought as well, where does CRISPR fit into nucleic acid chemistry? You can imagine, obviously, it would stabilize the RNA, and this was shown by the Matt Porteous lab with Agilent in 2015. They just put chemical modification. I keep bumping this thing. I apologize. I probably should just use the microphone up there. But they showed that you could stabilize the RNA by just putting some modifications on the ends. That made sense. But I really want to give a lot of credit to Ionis Pharmaceuticals and Don Cleveland's lab because they came up with this great sort of vision for how you might use chemically modified CRISPR RNAs for therapeutics. And it involves delivering the Cas9 enzyme by a virus, so a genetic, combined with a synthetic where you add in a chemically modified CRISPR, sort of like an ASO sort of treatment. And this allows you to really control the activity of the enzyme. So they published that in 2015. And I'll mention IDT has done a lot of chemical modification of CRISPR. It's not been published, but they sell this as their Alter CRISPR product online. Okay, so what I decided to do is I wanted to focus on CRISPR modification I worked in David Corey's lab, so I was familiar with nucleic acid therapeutics, but also wanted to kind of probe these enzymes and see what can they tolerate. It's an RNA-guided enzyme. At least the guide region should be able to tolerate pretty substantial modification. So we just started really simply with DNA. Why DNA? It's cheap. It has predictable properties, and it's easy to switch out. And so bottom line was we found that you have to keep RNA bases in or near the seed region, and there has to be a sufficient number of them. That's number one. Number two, if you put DNA bases in this region, in the 3' end, you can get dramatic improvements in the cleavage activity. We found that this held true for a couple sequences we tested. It also seemed to generally work for another related Cas9, Staph aureus. And when we tested it inside of cells, these also worked. Trends seemed pretty similar. So as a biochemist by training, I'm curious why we're getting this enhanced activity. And so we've done a little bit of biochemistry, mostly in vitro biochemistry, to try and figure out why is this helping out so much. So I won't go into details about this, but basically we saw that we're getting the same cleavage product. So when an enzyme works faster, sometimes it gets sloppy, so we wanted to make sure that wasn't a problem. And then we did a cleavage assay where we offered it either a perfect match or a mismatch target to see what was its mismatch discrimination, its ability to choose the perfect match over the mismatch. And we see that the DNA substituted one, called J in orange, had similar mismatch discrimination to a single guide RNA, and they both were over 2-fold better than an unmodified CRISPR RNA. When you did kinetics, enzyme kinetics, you could see that the DNA substituted CRISPR RNA works extremely fast. This reaction's done. It's actually very hard for my students to get the time points needed with the assay we were doing to actually fit this. And here's a single guide RNA. Single guide RNAs work with much, much slower kinetics, and that's because I have a couple reasons why I think that is, but you're fusing an RNA that evolved to be 2 separate RNAs with the protein. And it has to do with protein dynamics and its exchange or interconversion between on and off states. We also found that anytime you put DNA bases in there, you tend to improve the binding affinity of the CRISPR RNA for the Cas9. So you get order of magnitude improvement in your KD binding over an unmodified CRISPR RNA. This is even true for CRISPR RNA right here. This CRISPR RNA has no RNA bases in it. It's all DNA, so it's sort of a control. It assembles a beautiful RNP complex. However, it doesn't bind target DNA. So you get higher affinity and better assembly as you put in more DNA bases, but you also get weaker, lesser binding to targets. So based on these initial substitutions with DNA, we decided to go through and sort of test our hypothesis of A-form, maybe A-form-like structure in that seed region, which would explain why you needed RNA bases. And our goal was to put in DNA or RNA analogs or mimics into these regions to sort of probe that. So I don't expect you to actually read this. So I pulled out a couple examples, some here that worked okay, some that didn't work well, some that worked really well. And you can see the cell-based editing works pretty good also. I want to point out, this one right here has no RNA bases in it. We have a couple examples of no RNA bases in the whole CRISPR RNA. And general rules are you need that A-form-like structure in or near the seed region. You want to avoid bulky modifications. And DNA analogs are well tolerated outside this region, and you don't need RNA bases. So I think CRISPR is actually good for us because it shows us, whereas typically we want to avoid bulky, big payloads for therapeutics, we want to avoid that. CRISPR makes that pretty much unavoidable. You have to deliver a big gene, a big RNA, or a big protein complex. So because of that, and I think those delivery problems will eventually be solved, that really opens up the opportunities for us to envision new things. And for me, I'm going to think about novel RNA-guided enzymes that we can do new things in the cell with, or for new therapeutics. So hopefully you've had some time to think a little bit about what we've learned from RNA-guided enzymes, how they've helped us with therapeutics, and where we might be able to go with some of these enzymes, or even dream up new enzymes. And I'll just thank my students who worked on this. Really want to thank Masad Dama, who's designed and made all the chemically modified CRISPR RNAs. And we started a company called Guide BioSci so we could focus on CRISPR discovery and development for therapeutics and biotech and just funding. Thank you.
Thanks very much, Keith, for a very broad perspective on the field. Have you found any RNA modifications that can be tolerated in the seed region?
Yes, 2' fluoro is very well tolerated in the C region. It's one of the best replacements. 2', 5' linkages also, to some degree.
Okay, great. Lots more potential for discovery there. Any other questions? Okay, well, let me present you with Congratulations.